Hey class, in this discussion we are going to focus on the spinal cord. So let's dive into it. Here we can see a overall diagram of our spinal cord. Please note or focus on the fact that our spinal cord, as its name implies, is rather cord-like. We have a band of nervous tissue. This band of nervous tissue is mostly going to be white matter or consisting of axons, but surrounding it we're also going to have some layers of protective sheaths, and then we're also going to have signals entering and exiting the spinal cord as well. So as we're looking at our spinal cord, let's talk about functions. The functions of our spinal cord are multifaceted. For instance, we use our spinal cord for conduction. We use it to carry information to and from the brain. So we're going to carry sensory information up to the brain, motor information down from the brain. We also have some neural integration in our spinal cord. When we say integration, I want you to think of processing information. So we, in our spinal cord, our spinous neurons, are actually able to integrate or process information. Most of the processing of information in our bodies takes place in our brains, but there's a little bit of processing within our brain, within our spinal cord. For instance, we can have bladder control. Reflex arcs too are going to be processed within our spinal cord. So when you're stepping on that hot nail or stepping on that sharp rock on the ground and you immediately withdraw your foot and pull it back, that decision to withdraw your foot was made in your spinal cord. We didn't even wait for it to be sent to our brains. As we look at our spinal cord, it helps us with locomotion because we have central pattern generators. And when we think of these central pattern generators, they're just a group of neurons that are going to coordinate some repetitive sequences. So they are going to have some rhythmic depolarization and repolarizations to their action potentials to help modulate extraneous signals that come to those neurons. So they're going to resist inappropriate depolarizations or inappropriate action potentials to help give us a consistent pattern. We also are going to have reflexes modulated with our spinal cord. In particular, these reflexes are going to be in response to stimuli. I'd mentioned the sharp rock. We also have the classic burn your hand on a hot stove. We also have some posture as well, coordination and protection. When we think of protection, these are what most people think of when they think of reflexes. Coordination, think of how you reflexively adjust your arms and adjust your legs in response to losing your balance or having your center of gravity adjusted on you. As we look at our spinal cord, surface anatomy can be quite interesting. For instance, our spinal cord is, well, a cylinder of nervous tissue or a long tube of nervous tissue. It starts at the brainstem, particularly at the medulla oblongata, right where the brainstem exits the foramen magnum of the occipital bone in the skull. So the moment it leaves the skull, we recategorize it as the spinal cord as opposed to the brain. As we look at the spinal cord, it occupies the upper 2/3 of the vertebral canal. And I'm going to say this again differently. The lower 1/3 of the vertebral canal is not filled with the spinal cord. The spinal cord ends at the first lumbar vertebrae, L1, or just very slightly beyond that L1. I remember in particular, I found this to be quite interesting when I was a student because I just operated under the general assumption that your vertebral canal was completely filled with the spinal cord. It's not. As we look at our spinal cord, it's about 1.8 centimeters thick, 45 centimeters long, and I find this to be particularly interesting. This means that we have axons of neurons that are about 45 centimeters long, traveling the length of our spinal cord, particularly within the white matter of the spinal cord. We have 31 pairs of spinal nerves. Those 31 pairs of spinal nerves correspond to different vertebrae. We have our cervical, our thoracic, our lumbar, and our sacral vertebrae. And as we look at our spinal cord, it's going to be broken up into segments. Each part of the spinal cord, each region of the spinal cord is going to be supplied by a pair of spinal nerves. So here we have one segment, here we have another segment, so on and so forth. Oh, I went too far. And how do I go back? Give me a moment here. I can figure this out. I promise. I'm a smart person. There we go. Right click, previous, right click, previous. There we go. All right, we're back on track. So, as we're looking at the surface anatomy of our spinal cord, there's also going to be some grooves or indentations. These grooves or indentations can be fissures or sulci, depending on the depth of the groove. Generally speaking, we use the term sulcus for a shallow groove and the term fissure for a deep groove. This will become more pronounced as we get to the neuroanatomy of the brain. We haven't quite gotten to that point in this class yet. So as we're looking at a spinal cord, there can be grooves going the length of the spinal cord. This groove going the length of the spinal cord is going to be in both the posterior and the anterior. And as the name implies, we have the anterior median fissure and the posterior median sulcus. Here we have a posterior view of our spinal cord. So I just highlighted in green that posterior median sulcus. It's also going to be, the spinal cord is also going to be broken up into regions. So we have the cervical region by the cervical vertebrae. We have the thoracic region by, well, the thoracic vertebrae. And then down by the lumbar vertebrae, we have the lumbar enlargement. And this lumbar enlargement right here is going to be an expansion of the spinal cord to account for the fact that the lower 1/3 of the vertebral column is going to be filled with the cauda equina. The cauda equina in Latin literally means the horse's tail. Equine for horse, cauda for tail. And as we're looking at that cauda equina within the vertebral column, it's a bundle of nerve roots within the vertebral column that is resembling a horse's tail. Now, right after this lumbar enlargement, we have a fine point. The point itself is going to be referred to as the medullary cone. So let me shift colors here. That medullary cone is represented by the red triangle. And then let's go with blue. We have all those nerve roots coming down that represent the cauda equina. So as we look at the coverings of the spinal cord, the coverings are referred to as meninges. These meninges are 3 membranes. They're rather fibrous, and they are going to enclose most of the central nervous system, the brain and the spinal cord. And as we're looking at these meninges, they serve to be a protective barrier. They help to separate the soft tissue of the nervous system from the impact on the bones of the cranium or impacting the bones of the vertebral column. On the outside, we have the dura mater. And when we look at this, dura mater is German for tough mother. And as we look at the dura mater, it's here shown in kind of this orangish brown color. It's the superficial layer of the meninges. And it's very tough, it's very fibrous, and it's going to be separated by from the vertebrae by an epidural space. So when we think of epi, think of superficial to. And when we think of dural, think of the dura mater. So this epidural space is the space that is above or superficial to the dura mater. Deep to the dura mater, we have the arachnoid mater. Arachnoid is the root word for referring to spider, so it's the spider matter. And it is called the arachnoid mater because it has fibers that span through the subarachnoid space. And those fibers are branch-like, they're filamentous, and they resemble a spider's web. So because the fibers of the arachnoid matter resemble the spider's web, we call it an arachnoid matter. So as we look at the arachnoid matter, it's shown here in kind of the off-white blue. And this arachnoid matter helps to take the dura mater and adhere it to the underlying pia mater. When somebody has a lumbar puncture or spinal tap, spinal tap or lumbar puncture is going to be putting a needle into the subarachnoid space. And while that needle is in the subarachnoid space, it's going to remove cerebral spinal fluid, a separate liquid that's circulated throughout the central nervous system. I'll often refer to cerebral spinal fluid as CSF, just to save some time. Now, as we're looking at the pia mater, this is the deepest layer of the meninges, and this pia mater contains a very delicate membrane. It's shown here in pink, and that pia mater is very difficult to dissect away from the underlying nervous tissue. When I do sheep brain dissections with my students, this pia mater is almost never dissected away just because we don't have time for that. Ain't nobody had time for that. So as we're looking at that pia mater, it's going to follow the bumps, the grooves, the sulci, the gyri, the fissures of the central nervous system, particularly in this context, the spinal cord. And it's very fibrous. As it heads inferiorly, it's going to reach the cona medullaris. So as we're getting to the inferior cone, let me insert a whiteboard here. So as we get to that inferior cone, we have the lumbar enlargement. And then after the lumbar enlargement, we have the cona medullaris. And then after the cona medullaris, we have that pia mater wrapping around. And at that pia mater, as it wraps around, it eventually gets to the terminal phylum. And that terminal phylum is going to just be at the tip of the cona medullaris. And then we're going to have a long string of pia mater, or fibers of the pia mater. And that long string of fiber is going to be referred to as the coccygeal ligament. Let's clear the screen here and progress. So that's all we have for our recording today. If you have any questions, please feel free to post them on the class discussion board or shoot me an email. And as always, happy studies.